Great. Welcome, everyone. We are honored and excited to host today's MSQ Webinar Series with our special guest speaker, Dr. Kwon, CEO and founder of AUTOTAC from Korea, innovative drug platform based on targeted protein degradation. And we are really excited to start off with getting to know a little bit about Dr. Kwon and the history to date before he makes a presentation, and we'll also field questions from the audience. Just to give you a little bit of background, AUTOTAC Bio is a new drug development company based on protein target decomposition platform technology. It's a next generation new drug development platform technology. We are very excited to learn more about that. Let me start with introducing Dr. Kwon. He's the founder, as I mentioned, CEO. He has deep insights in the research field of protein degradation, specifically in the relationship between N-degron and autophagy pathway. With the knowledge of the role of N-degron pathway in cancers, neurodegenerative disease, and rare incurable diseases, Dr. Kwon and his team developed targeted degradation platform targeting pathogenic proteins, the world's first P62-based TPD platform. Dr. Kwon, it's a pleasure having you on our webinar. Nice to have you.
Okay, thank you very much for your invitation to this seminar series.
Great. Maybe we can start a little bit with your background. Tell us a little bit about yourself, how you got to be the CEO and founder of this cutting-edge technology platform and novel and innovative therapeutic.
Okay, so I have been working on the ubiquitin proteasome system and the autophagy lysome system. Basically, I have been working on the cellular degradation systems in over 30 years, since the early 90s. And since then, I have been actually studying the principles that modulate, especially the targeting and the selective recognition of cargos destined to proteasome or lysosome. And we recently worked on the autophagy system, and the autophagy system has been thought it's non-specific Burki degradation systems, and we found surprisingly that the autophagy is selective enough and also very accurate. So also we realized that as opposed to ubiquitin proteasome system, where 600 different ethyl ligases split their jobs to ubiquitinate for degradation, but in autophagy, the receptor P62 is responsible for almost 70% of all autophagy flux for protein aggregates and many other proteins and also types, other cargos. So we naturally worked on the regulation biochemical mechanisms of P62 over 20 years and end up with the physiological ligands, and we converted to chemical moiety small molecule ligands that selectively recognize, conditionally activate P62, and later we advanced this small molecule chemicals into Chimera, now called the AUTOTAC Bio. I will introduce our technology.
Great, great. You've also published around 140 papers. You achieved an H index of 48. You filed around 80 patents. How do you find the time? Where does this passion come from?
That's a very difficult question. I devoted to science since mid-20s, and so most of my life actually was spent for studies in ubiquitin proteasome system and autophagy, and investigation is actually my pleasure, so it's my hobby as well.
Interesting. I also read in your bio that you serve as president of the International Society of Protein Termini, ISPT. Can you tell us a little bit about that?
Yes, that's I have been working on protein degradation in my field, and years ago I realized that many scientists in Europe and the United States and Asian countries share the common interest in the protein degradation. So I proposed to make a non-profit cooperation and so named ISPT. And now biannually we hold the international conference starting from Seoul, Korea, and Bergen, Norway, and next year the Oxford in England. So this is how. So we basically this is the international network of scientists interested in protein degradation.
Interesting. Yeah, so you've been spending over 30 years on fundamental impacts in degradation biology and drug development to treat otherwise incurable diseases. We really are honored and excited to learn a little bit more. Maybe you can share your screen and present AUTOTAC and your company, and then we can field some questions on the back end of your presentation. Thank you.
Okay, thank you very much. Do you see this screen?
Yep, perfect.
And okay, so today I will just focus on the technology itself. So I have been working on the protein degradation systems, mainly in the ubiquitin proteasome system and the autophagy. And in the cellular degradation system is composed of mainly ubiquitin proteasome system and autophagy. These are two major systems responsible for degradation of the proteins. And in ubiquitin proteasome system, approximately 600 different E3 ligases induces ubiquitination of substrates for proteasomal degradation. Before degradation, the targeted substrates are unfolded and fed into internal cavity of proteasome, but this proteasome inner diameter is as narrow as 13 angstrom, while the proteins are 50 or 100 angstroms. Therefore, they must be unfolded, and many diseases are caused because these targeted proteins are not properly unfolded, such as Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS. And autophagy use lysosomal hydrolases to target various cellular cargoes, and these cargoes are sequestered by autophagy membranes. Through autophagism, they are eventually degraded. Ubiquitin system and autophagy crosstalk each other. In general, the proteasome targets native proteins and soluble misfolded monometic proteins. And once these proteins are aggregated and cannot be degraded by ubiquitin proteasome system, they are collected mainly by autophagy receptor P62 and sequestered by autophagy membranes and degraded by lysosomal hydrolases. Over 20 years, we have been working on the selective autophagy system, how the cargoes are selectively recognized and degraded by lysosomes. And we were surprised that the autophagy system is selective enough and accurate enough. So basically, they can selectively recognize like a ubiquitin proteasome system and degrade. So also, we found that the P62 is major autophagy receptors amongst the 20 or 30 different autophagy receptors, especially for protein aggregates. So we started on the autophagy protein degradation and how these cargoes are selectively recognized and degraded. And I'll skip the details, but this all represent physiological ligand. And we found that the oral moiety, physiological ligand exposed from the proteins basically induces autophagy and actually induces degradation of autophagy cargoes. And we found that this R binds P62, inducing conformational change. So P62 is normally inactive and closed, and upon binding to physiological ligand through specific hydrogen bonding, P62 undergo conformational change, open configuration, and these domains induces the self-polymerization in complex with cargoes, leading to targeting to autophagy membranes and lysosomal degradation. And we also learned that this physiological ligand bound P62 migrated to endoplasmic reticulum to induce more biogenesis of autophagy membranes. And later, we use a 3D modeling and virtual library screening of 500,000 compounds to convert this physiological ligand R to chemical moiety. We call it autophagy-targeting ligand, ATL. And we learned that this ATL forms a specific hydrogen bonding with the specific residues of P62. And we performed the crystallography of P62 for subtle optimization at the atomic level. And eventually we ended up with small molecule ligands that can modulate now autophagy proteolysis of aggregates. And we learned that this ATL itself was not enough to induce strongly the degradation of protein aggregates. So we now converted the chimeric configuration. So this first drug binds target, and second drug ATL binds P62, inducing conformational change, leading to polymerization in complex with targets and autophagy targeting and autophagism and lysosomal degradation. And the chemicals are recycled back to a new cycle. And so I will introduce AUTOTAC technology. In central dogma, DNA contains the genetic information which is transcribed into RNA and translated into proteins. And DNA can be selectively edited using DNA editing technology. And RNA can be selectively degraded by siRNA, and the proteins, intracellular proteins, can be selectively degraded by either proteolysis targeting chimera through ubiquitin proteome system and autophagy targeting chimera through autophagy. And AUTOTAC is basically first-in-class the TPD technology versatile for almost all the intracellular proteins. And because we use lysosomal hydrolases to degrade the proteins, we naturally, for now, focus on CNS diseases, such as Alzheimer's, amyotrophic lateral sclerosis, progressive supranuclear palsy, and Parkinson's. And amongst them, AD, ALS, and the PSP is now completed the GLP tox studies, and we are scheduled to enter phase 1 and phase 2 clinical trials. And we also have other pipelines. And so this year, we will apply for IND this week, next week. And so we plan phase 1 study for Alzheimer's disease next year. And P2 study to 2025, and progressive supranuclear palsy phase 2 after P1 study, and also amyotrophic lateral sclerosis ALS phase 2 study after P1 SAD study. And so neurodegenerative diseases affect 100 million people worldwide, including AD, PD, HD, ALS, and PSP. And these diseases represent a major threat to human diseases. And we have many patents, approximately 20 different patents that form IP network of AUTOTAC technology. These are individual patents. And so I will first talk about Alzheimer's disease affecting 42 million people worldwide. And the prevalence is ever increasing. And we developed oral drug that induces targeted degradation of pre-existing tau tangles in the neuron. And so we chose intracellular tau tangles as opposed to many other companies focusing on extracellular A-beta, because tau tangles are directly responsible for the toxicity to neurons. And this candidate compound, Chimera, as a molecular weight is around 624, and induces degradation of tau aggregates at low nanomole, and no off-target degradation from monomers, and in vivo efficacy in mice, cognition, behavior, locomotive activity, tau degradation at all 1-MPK, PO, BV penetration 10%, NOAL, rat 240, dog 500, and other toxicity profiles are excellent. Safety margin is over 100, and phase now we are entering clinical trials. These are some scientific data. Because of time limit, I'll skip the details. So this is DC50 low nanomole, and induces degradation of tau aggregates, but not the monomers. Same data. And you see this fluorescence represents tau aggregates degraded by AUTOTAC compound, functional complementation. Protein mix of 15,000 proteins show no significant degradation of target degradation of cellular proteins. And in mice, RTG45110 overexpressing mutant tau P301L, and we used the 1-MPK and 10-MPK oral administration. This represent tau tangles from the brains which are degraded by 1-MPK. You can keep in mind that NOAL in dog is 500 MPK, so you can imagine that the safety margin is huge. And this show the different forms of tau, possible taus, which are degraded from the affected brains, similar results. And this basically the tau tangles are degraded. Fluorescence represents tau aggregates in the brain, which are reduced by 1-MPK, the PO. And this is open field test, and you see this is crazy hyperactivity, which is normalized by 1-MPK drug. And this is a novel. This is let me see, open field test. This is also novel object recognition test showing the efficacy at 1-MPK Morris maze analysis, actual the data. I will skip the details. And this is another mouse model expressing tau P301L by FC. Now we increased the dose to 20 mpk and reduced the doses 8 times. And so we see still the degradation of tau tangles. These dirty signals, fluorescent signals, represents tau tangles which are eliminated by oral administration, now 8 times, open field test. And this is a typical tau tangles in the brain, which are cleaned up by oral administration. And we see here the hinder leg is shows abnormal behavior. Usually rodents spread the hinder legs when they are grabbed by the tail in preparation to fall down on the floor. And so upon administration, they show the relatively normal behavior. And this is typical walking pattern at the later stage. So in the later stage, the motor neuron also is affected, and they cannot properly walk. Actually, they swim on the floor, and this drug basically shows a dramatic therapeutic efficacy. And this is ANL is actually target binder used to recognize tau, and this itself is actually phase 2 clinical trials for Parkinson's disease for its ability to bind and slow down aggregation. But this is a 500 mpk clinical dose, and at the 1 mpk, when this same compound is applied to AUTOTAC, it shows much stronger efficacy even at 500 lower dose. And these are GLP tox studies, NOAL in rat and the dog, and other toxicity profiles, and safety margin is over 100. And we are planning to do the P1 study at the Seoul National University Hospital in Korea. And this P1 study plans. And this is about PSP. PSP is progressive supranuclear palsy belonging to tauopathy. Tauopathy actually has so many different diseases. All these are all different diseases, and naturally, the tau degrader can be applied not only for Alzheimer's disease. This is secondary tauopathy driven by tau and amyloid beta. The PSP is a more direct actually indication that can be, that we can apply. So this PSP is different in symptoms from Alzheimer's disease, and in Japan alone, there are 20,000 patients registered, and these patients actually die within 5 years or so after diagnosis. So this patient, the doctor says that every time she sees the patients, patients are dying without drug. So we are basically very urgent to perform phase 2 clinical trials for PSP patients. And this is amyloid amyotrophic lateral sclerosis. This is a representative the motor neuron disease affecting actually many people. And ALS patients die usually 2 to 4 years after diagnosis. And so far, approximately 400,000 patients are alive. But every year, same number of patients die actually. This is a very serious disease with no drug at all. And so we target TDP-43 aggregates, which are common in the 97% of all ALS patients, and also the half of the frontotemporal dementia and even the Alzheimer's disease. We have two compounds, and this ATC 141 is now entering the phase 2 clinical trials. These are some scientific data or skip, and this mouse model of ALS overexpressing mutant aggregation-prone TDP A315T, and so we see degradation of TDP-43 aggregates. This is insoluble aggregates from the spinal cord, and this is the hinder leg and the forelimb, the strength, muscle strength, and this is the ALS mouse model. You can see that the hinder leg shows different abnormality. Usually, the rodents basically the hinder leg should be positioned within the body line, but they're outside. This is a typical phenotype. And so we, upon oral drug treatment, 10 mpk and 24 times, only 8 weeks, and we see dramatic therapeutic improvements in ALS. This means that we can actually cure this, not just delay the symptoms, but we can cure the ALS, the patient. And this is another model, and we see that ALS mice are impaired in neuromuscular coordination, which is greatly improved upon the oral treatment of AUTOTAC degrader. And so we see a huge therapeutic actually improvement. So we can cure this drug disease. And Parkinson's disease, we just published this work. So Parkinson's disease is basically caused by the alpha-synuclein aggregation and in the brain. I'll skip the pathogenesis, and Parkinson's disease was discovered actually 200 years ago. Still, there is no drug. And all the drugs so far available are symptomatic. And so our drug is disease modifying, fundamentally eradicate the pathogenic agent from the brains patients. PD affects more than 10 million people around worldwide. Our skip science, and these are some science. And so we basically see that our drug induces degradation of alpha-synuclein aggregates in the site of injection, and also the opposite, the hemisphere of the brain associated with the increased, the improved locomotive activity. So this was now just published the last month. So basically, we are a small company, and right now we are working on mainly neurodegenerative disease, but the same principle can be applied for many other diseases and targets. So I'll take some questions.
Great. Thank you very much for that presentation. Very enlightening. Learned a lot, and looking forward to addressing some questions from the audience. Give me 1 second. Here is one. How would ATC 102 differentiate itself from other drugs currently under development for AD in terms of efficacy and safety?
As you know, a few companies now developed antibody drug that can delay the pathogenesis of Alzheimer's disease by 27% or 35%. This means a few months. So these available drugs target extracellular amyloid beta, leaving the intracellular tau tangles intact. And so our degrader eliminate fundamentally the intracellular tau tangles. Therefore, we believe that our degrader can actually improve, treat, cure Alzheimer's disease, at least partially. So that's the major difference. Our goal is not simply delay the symptoms. We believe our degrader can at least significantly cure the disease.
Great. The drugs, here's another one, because I think you answered the question about that. The drugs have functioned preferentially better in late stage p-tau species. What's your expectation for its efficacy for early stage AD?
Our data shows that actually our degrader can induce degradation of both early stage phospho-tau species as well as later staged phospho-tau species, slightly preferentially toward the later stage. Therefore, we believe that our degrader can be applied to the later stage Alzheimer's patients as well as early stage as well.
Great. Here's another one. Why was P62 chosen as the autophagy receptor out of the other receptors?
In autophagy system, there are more than 30 receptors now known to function. And as for degradation of aggregates, approximately 20 different receptors are known to be involved. And over 20 years of research, we actually, from 10 years ago, we already recognized that P62 is responsible for roughly 70% of all autophagy flux for protein aggregates. Therefore, it is natural that P62 is just a bottleneck in the master regulator in autophagy degradation.
Thank you. How does AUTOTAC approach the misfolded protein compared with PROTAC and other autophagy-based degraders?
PROTAC use proteasome, and which can actually target monomeric proteins and soluble misfolded proteins. But because of physical barrier of proteasome, the PROTAC cannot target beyond that, which means oligomeric species and pre-existing tau tangles. And AUTOTAC use, so in that case, PROTAC is more like a preventive medicine. By degrading monomeric species, it prevents from the accumulation of tau tangles. But when the patients visit a hospital and the patients recognize the disease, already their brains are full of the tangles and aggregates. And we have to eliminate pre-existing large size aggregates from the neurons. And because AUTOTAC use lysosomal hydrolases, we can basically eliminate existing tau tangles quite effectively and safely.
Great. One challenge of PROTAC is low affinity between affinity target, binding ligand, TBL, and target. How will P62-based ATPD approach those low affinity target binding ligand, TBL target combinations?
So one major difference between PROTAC and AUTOTAC is that PROTAC targets the proteins and induce ubiquitination through one-to-one interaction. So one E3 binds one target molecule and induces ubiquitination and complete that job on site. But in case of AUTOTAC and the PCK2, upon binding to the ATL, PCK2 forms self-oligomerization in complex with targets. This self-oligomerization induces the multivalent interactions. Therefore, affinity versus avidity is basically the avidity is increased 100 times, so 1,000 times compared with the one-to-one interactions. You can imagine that the zipper, just a small one zipper is very weak, but if we zip the long zipper, then it's quite tight. And basically through this multivalent interactions and it's a synergistic interactions, the cargos are basically effectively targeted at the low nanomolar range.
I think we have time for one more question. Ubiquitin-dependent PROTAC is largely confined to a limited set of target proteins and of E3 ligases. Compared to E3 ligase, how is P62 act as a pan-autophagy receptor?
As I mentioned, in ubiquitin proteasome system, the 600 E3 ligases split the job of a whole ubiquitin proteasome system, and each of E3 ligases must form tertiary complex with the substrate. Different from PROTAC, PCK2 basically is activated through intramolecular conformational change, independent of the target. Therefore, the PCK2 does not need to form tertiary complex with the targets. Therefore, by simply changing target binder, we can actually target most of given intracellular proteins. This is the major difference between PROTAC and AUTOTAC.
Great. Well, I think those were most of the questions. I know we have to unfortunately end the webinar. We learned a lot. Dr. Kwon, any final words you want to say before we close out the webinar?
Yes, and so we are a small company, recently established and relatively not known well in the global market. And I hope this seminar give an opportunity to introduce our technology to a more global market. And so we are actually open to the licensing out global or regional, and as well as equity investment. And indeed, some of our assets are now under due diligence with the top 20 global companies and also top 30 one company. We are scheduled to conclude around 5 to 6 months from now on. And so we have a window of time to discuss this licensing out or equity investment. Thank you very much for your attention.
Great, thank you. Again, we're very excited and honored to have you. Thank you for sharing your knowledge and for us to learn about yourself, your company, and all your achievements today. Congratulations. We look forward to more novel achievements from your side in terms of your platform and your assets. Just as a word of, I guess, from MSQ side, we are, because this surge of targeted protein degradation drug development, we are excited in terms of, growing and evolving with this space. So we do have a webinar scheduled for August 10th and August 24th, part 1 and part 2. We are inviting MNCs that are heavily invested in TPD, emerging global players and deal makers. So we're very excited about that. Please tune in for that in terms of those webinars coming up, panel discussions. Again, Dr. Kwon, thank you very much for your time tonight. Really appreciate
Thank you very much, the MSQ team.
Great. Thank you so much.
Okay,
Pleasure.
bye. Yeah.